Okay, so our last presentation in this series is Erica Whitney. Could mycorrhizal fungus colonization of red raspberry reduce pressure from root lesions?
Okay. Oh, there it is. So, hi, my name is Erica Whitney. I am a master's student at Western Washington University, doing research in Dr. Rebecca Bunn's soil ecology lab. And our project investigated whether mycorrhizal fungi available in raspberry fields or other sources had the potential to influence the symptoms of root rot and root lesion nematode on young raspberry plants. This project was a collaboration with Dr. Lisa Deveter's lab down at WSU. If you were here yesterday, you might have seen Chen Win's talk on some commercial brands of mycorrhizae and their influence on raspberry nutrition. So today I'm only going to be presenting some of the preliminary results of our experiment. We did let it run a little bit longer so that those growth responses could fully develop, but that means that we are still waiting on a little bit more data to come in and for a little bit more analysis. So here's some of our results up front. Don't worry, I'm going to be talking about these in more detail later on. I just wanted to give you a snapshot. So first off, we did find that we had high levels of colonization resulting from all of our mycorrhizal treatments. That includes one that was just soil straight out of a Whatcom County farm, which suggests that there might be ample mycorrhizae available in your field soil already. We saw that there was an effective mycorrhizal fungi. They had an increased leaf content for plants that were treated with mycorrhizal fungi. And those treatments also either didn't affect plant height and biomass, or they slightly reduced height and biomass. The pest treatments had a neutral to no effect on plant biomass or plant height, whether or not there were mycorrhizal fungi. And so this might be because of environmental conditions in the greenhouse that weren't necessarily conducive to those pests. So let's take a step back, and we're just going to talk about why we're interested in soil ecology of agroecosystems. Soil ecology is really useful for understanding and improving the sustainability of our agricultural systems, because plants have evolved over millennia to rely upon beneficial biota in the soils, including fungi and bacteria that help to support their nutrient acquisition, their pathogen protection, and more. Of course, in those soil communities, there's also harmful pests, so pathogens and root herbivores that are detrimental to plants, and also organisms that maybe don't directly interact with the plants, but that are important nevertheless, like decomposers. And in the soil, all of these organisms are interacting with each other all the time. And so it's been shown that in natural systems, the diversity of microbiota present in the soil can help to reduce pathogen pressures via direct competition with those pathogens and also through other mechanisms. So building on this understanding of, we do know that standard agricultural practices have an effect on the balance of beneficial and detrimental organisms in the soil. In intensively managed systems, what we see oftentimes is that there's a reduction in many of the plant beneficial species, as well as an increase in crop pathogens over time. Dr. Pierre Mariotte published a really great review on this subject, writing that research into the role of beneficial microbiota in agroecosystems will offer a suite of tools to improve the security of our food systems by promoting long-term plant health while minimizing the environmental impacts. So some of the tools that he was discussing that have shown promising results include adoption of cultural methods that can help to promote soil microbial diversity, and also the reintroduction of key plant beneficial microbiota into your systems. So our study falls under the second of these two strategies. The key beneficial biota we were interested in are arbuscular mycorrhizal fungi. I'll also refer to them as AMF to make it shorter. These fungi form a relationship with the roots of plants. The fungus depends on the plant for carbon, so up to 10% of a plant's carbohydrates produced through photosynthesis can get shuffled down to the fungus, and in return, the fungus grows out into the soil and is able to access nutrients in the soil that are otherwise unavailable to the plant, and it can send those to the plant. In addition, and one of the nutrients that AMF are particularly known for enhancing plant access to is phosphate, which is limiting in a lot of soils and is also a problematic pollutant when it enters waterways. So in addition to improving plant nutrition, AMF have been studied in both natural and crop systems to improve stress tolerance of plants to things like drought and salinity, and also to reduce the effects of pathogens on plants, both through direct interaction with those pests and also by indirectly altering the plant's defense responses. It has been shown that protection is more effective when the mycorrhizal fungi are introduced before the plant is exposed to those pathogens. So that being said, this is not always a net beneficial relationship. So it can range from positive to negative, and that depends on both the fungal species involved, the plant species, even the genotype of plant that you're using, and also the environmental factors. It's only beneficial as long as the benefits being offered to the plant exceed the cost of those carbohydrates that the plant is giving the fungus. So with this in mind, we're going to be focusing on mycorrhiza of raspberries. Here in Washington, the productive lifespan of raspberry crop plantings has decreased over the last decade. Two known culprits afflicting the roots of raspberry are Phytophthora ruby, which is shown in the upper photo. That's an oomycete commonly known as root rot. And one study by Dr. Jessica Gigo found Phytophthora ruby in 10 out of 10 fields sampled in Skagit and Whatcom counties. So it's really common in this area. And another common pest is Pratylenchus penetrans, or the root lesion nematode. This is a migratory nematode that feeds on the fine feeder roots of plants and exists in the soil as well as within plant roots and can stunt plant growth. And so despite intensive fumigation practices to try to control these and other pests, the downward trend in raspberry field productivity still continues. So this has piqued a lot of growers' interest into alternative sustainable soil management strategies. And to connect these pests back to mycorrhizal fungi, we have seen in studies of other crop systems that mycorrhizae can protect plants from other Phytophthora species, and that prior colonization by AMF can improve establishment rates in Pratylenchus penetrans infested fields. So with this in mind, we were wondering if mycorrhizal fungi might be a key player for raspberry plants capable of improving their resilience to these two pests, and whether they might be lacking in raspberry fields. So we started by looking at roots associated with an ongoing project up in Whatcom County, where a single farm had distinct areas of high vigor and low vigor raspberry plants. We obtained roots from each of those vigor categories to assess whether there was a difference in rates of colonization in those roots. In order to assess mycorrhizal colonization, we clear the pigment from the plant roots and then stain them with the compound that colors the fungus darkly so that we can look at them under a microscope. The picture that you're seeing up here is a cleared and stained root at about 400 times magnification that shows some of the structures we're looking at whenever we quantify mycorrhizal fungi. So first off, you'll see arbuscules. Those are the structures where the nutrient exchange between the plant and the fungus actually occurs. Vesicles, which are like a storage compartment for the fungus, and the hyphae, which is always going to be attached to these and is actually the primary structure of the fungus in the soil. And so what we found is that there were differences. In the low vigor areas, only about 50% of the root samples assessed had mycorrhizal fungi present, Whereas in the high vigor areas, that 90% of the root samples had mycorrhizal fungi in them. So this was interesting, and in our project, we were left with two major questions. First off, how will raspberry plants respond to colonization by mycorrhizal fungi? And not just will they respond to it, but also how might the differences in mycorrhizal fungi available in agricultural soil compared to unmanaged or wild soils affect those plant responses? Our second question was, how do mycorrhizal fungi affect plant susceptibility and responses to some common pathogens or common pests? And so to answer these questions, we designed a two-phase experiment. In phase one, we grew 40 plants in each of 4 mycorrhizal treatments. So we had 160 plants total. Those plants either were a control, which received no mycorrhizal inoculum. They could have received soil from a Whatcom County raspberry farm to represent our agricultural soil. That was an 8-year-old planting of raspberries in sandy loam soil, and the raspberries were Wakefield. Or soil from a thimbleberry patch to represent our unmanaged soil blend, or else a known community, which was what we would consider a rich community, which had 8 different species of mycorrhizal fungi in it. So for phase one, we started with 700 milliliter deep pots filled halfway with a sterilized soil mix, to which we added 30 milliliters of our inoculum source. And then we topped that off with more sterilized soil and a raspberry plant plug. And we grew these in phase one treatments for 10 weeks and then introduced our phase 2 treatments, which were the pest. So plants could either receive no pest, Phytophthora of ruby, Fratalentris penetrans, the root lesion nematode, or both. In phase 2, we took 1,000 milliliter tree pots and filled them with that same sterilized soil mix. This time that soil was either not inoculated or it was inoculated with Phytophthora ruby that we had cultivated in our lab at 10% by volume. Into that, we planted those 10-week-old plugs of raspberry that had been growing in their mycorrhizal treatments and topped it off with a little bit more sterile soil. For the nematode treatments, we extracted nematodes from the roots of established raspberry plants, and we injected those into the soil so that we could reach a density of 1,000 nematodes per pot. And what we saw is over the course of the experiment, we measured above ground features every 2 weeks for the entire 24 weeks. So this is a pretty busy plot over here. But the main thing that I want you to take away from it is that over time, the differences in height increased. So you see that they're really tight together at week 10, and then they get bigger further out. This is a busy plot, so to make it easier, we have the bar plot to help us take a look at this. On the bar plot, on the y-axis is the plant height in centimeters at week 10, and on the x-axis, we have our mycorrhizal treatments. The red bar represents the control, which received no mycorrhizal inoculum. And so what we see is that there were no differences in height across any of the treatments at week 10. To compare that with week 24, we started to see some differences. So by week 24, only one mycorrhizal treatment was significantly shorter. In this plot, the letters, if a treatment has the same letter, it's not statistically different. So these treatments here are not significantly different from one another, whereas the thimbleberry mycorrhizal treatment did show reduction in height by week 24. On this plot, it's again in centimeters, but we have zoomed in the y-axis, so note that it does start at 40 centimeters to make it easier to see. The control is about 145 centimeters or 57 inches tall, while the thimbleberry treatment was on average 7 inches shorter than that control. These differences in height are reinforced by the biomass data. So what we saw was that the bar plot on the left shows biomass in grams dry weight, and like height, biomass was only significantly reduced by the thimbleberry treatment. That's that purple bar here. In contrast, there was a positive effect on foliar nitrogen content by every mycorrhizal treatment. And that is the plot that's on the right. So what we want to take out of here is that for only one mycorrhizal treatment, the increase in foliar nitrogen was paired with a reduction in biomass, whereas for the other two mycorrhizal treatments, that increase in foliar nitrogen had no effect on biomass. So in answering our second question, this is the one that we still have a lot of data coming in for and more analysis to do. What we've observed so far is that there was no effect on biomass by our pest treatments across all mycorrhizal treatments. This could have been due to environmental conditions like high summertime temperatures in the greenhouse that aren't conducive to those pests. It's also partially explained by some qPCR results from the USDA and Corvallis that suggests that for the small amount of samples that we sent them to look for Phytophthora infection, they did not detect Phytophthora in any of those samples. So that suggests that we did not have successful infection by the Phytophthora. We do have results on the nematode density that we're just starting to analyze. And so even though, well, I guess I'll introduce this first. So on this plot, we have the nematode density for within the roots in nematodes per gram dry root. And those are broken out by mycorrhizal treatment again. The red bars again are the control, while the other colors are the mycorrhizal treatments. On the left side, you see that the ones that are without Phytophthora present, and on the right side, you see those that did receive Phytophthora in their soil. And even though there wasn't successful infection by the Phytophthora, we see an effect by this Phytophthora treatment. Overall, there was a reduction in the average density of nematodes in the roots when plants had also been exposed to Phytophthora, and there was also a reduction in the standard error on those nematode densities. So we're interested to connect this back to our earlier discussion of soil ecology, whether or not this might be indicating some kind of interaction between those two pests going on in the soil, whether this might be because of altered plant defenses due to that earlier exposure to the Phytophthora, or if it could be just that there were different abundances of the types of roots that the nematodes prefer, like the fine feeder roots, because of that exposure to Phytophthora. So we have a lot more questions about that we're going to be analyzing further. Also, other data that we're waiting on analyzing that'll be coming up soon is plant nutrient acquisition. We're waiting on macro and micronutrient content of the plants so that we can find out if there were any differences in the ability of the plants to take up nutrients, and also to finish assessing data on the degree of colonization. Right now, we've only assessed about 30% of the 160 plants for how much mycorrhizal colonization there was in them. And so once we have that full data set, we'll be able to look more at correlations between the degree of colonization and other plant or nematode responses. So again, a quick summary of what we have found so far. First off, that there was high levels of colonization across all of our mycorrhizal treatments, including soil from one of the raspberry farms here in Whatcom County. And that mycorrhizal fungi had a positive effect on leaf nitrogen content and either a neutral to negative effect on plant height and biomass. The pest treatments resulted in no effect on plant biomass with or without the mycorrhizal fungi. And we're still waiting on more data to receive and analyze that's going to help us assess some of those other responses. So with that, I'd like to thank the Washington Red Raspberry Commission and Western Washington University for their funding. A special thanks to Dr. Rebecca Bunn and Dr. Lisa Deveter, who had a huge role in this project. The Grower Cooperator Farms, who provided soils, roots, and support for the project, and the numerous other experts and volunteers who have been involved. I'm not sure how much time we have left, but if anybody has any questions.
We can take a few questions. Okay, two questions.
On your earlier photo, in fact, there's a couple of photos back at the greenhouse.
Yeah.
Is that right there? Is that because of a moisture problem, or is that deficiency of magnesium on that?
So these were the plants right before harvest at week 24. We did stop watering them right prior to harvest. So for about 1 week before harvest to facilitate being able to get things out of the roots or to shake off the soil from the roots so that we could assess them later. So these were our healthy plants before harvest.
Okay, and was there any difference in the pH in any of the tests, your soil pH, of making a difference to your test?
We have not assessed soil pH for what resulted at the end of all the different treatments. We've saved soil for each of the different 16 treatment combinations, but we haven't assessed soil pH yet. Are there any other questions?
Okay, looks good. Thank you.
Thank you.
Again, for